Alors bonjour à toutes et tous. Je tiens tout d'abord à vous remercier d'être venus en si grand nombre pour notre conférence conjointe CEF-IBIS. Aujourd'hui, nous avons le plaisir d'accueillir le professeur Nicolas Corradi. En passant, je crois qu'il y a eu une petite erreur dans la traduction anglais et français. Quand on dit associate professeur, ça veut dire l'équivalent ici à Laval, professeur agrégé, pas professeur associé. Bon, alors, Nicolas a terminé ses études de doctorat à Lausanne, à l'Université de Lausanne, en 2006, en Suisse, sous la direction, je crois, de Ian Sanders. Est-ce que c'est bien ça? Oui. Oui. Alors, il a été engagé comme professeur à l'Université d'Ottawa depuis 2010. Et dans son labo, il travaille sur deux groupes de micro-organismes dont il va nous parler certainement cet avant-midi lors de sa conférence. Il y a d'abord le microsporidies, qui sont des parasites intracellulaires qui vivent chez les animaux comme chez les humains aussi. Et là, évidemment, son labo a fait de percées quand même très intéressantes. Et ses travaux sont d'une notoriété internationale, publiés dans de prestigieuses revues comme PNS, Current Biology, eLife. Et d'ailleurs, en 2010, il a été invité à faire une revue de littérature sur ça, qui a été publiée dans Annual Review of Microbiology. Et finalement, l'autre groupe de champignons sur lequel il travaille sont les mycorhizes. Alors, je suis content de voir nos aînés, notre papa, sur les mycorhizes, André Fortin, Yves Piché, qui ont formé autant de jeunes Québécois, des Canadiens dans ce domaine-là. Bon, il y en a bien d'autres, mais merci d'être là. Et je sais qu'il y a aussi d'autres professeurs retraités. Je vois Alain qui est là aussi, mais il m'a dit aujourd'hui qu'il n'est pas encore retraité. Il continue à travailler. Bien, donc Nicolas travaille aussi sur ces champignons. Un domaine si passionnant. Moi-même, je fais ma maîtrise sous la direction d'André. Un domaine vraiment très passionnant de la vie à symbiose de plantes. Et jusqu'à présent, on a toujours, les chercheurs ont eu beaucoup de travaux sur ces champignons-là, mais la génétique de ces champignons reste quand même très difficile. Et dans les labos de Nicolas, ils sont en train de montrer un peu la génétique, probablement mendélienne, de ces champignons-là. Donc aujourd'hui, il va nous parler de la génomique évolutive de ces deux groupes de micro-organismes, le microsporidies et les champignons mycorhiziens arbusculaires. Donc, sans plus tarder, je vais laisser la parole à Nicolas, comme il est dans une université bilingue. Nicolas is a perfect bilingual, so feel free to ask your question in English, in French.
Or in Italian.
In Italian. Okay, so it's up to you, Nicolas. Okay. Thank you so much for accepting to come to visit Laval. Thank you.
Thank you. So, I've been told I can do it in English, right? Okay. Well, thank you very much for inviting me. It's a pleasure to be here. It's the first time that I come to the University of Laval, so it's a real pleasure to meet all of you. So today I'm going to talk about an interesting group of organisms, actually two interesting groups of organisms. I'm going to show you some of our past research on microsporidian and some of our recent breakthrough in regarding the genetics of the arbuscular-mycorrhizal fungi. So the first thing I would like to start with is this. So I come from Ottawa, which means that about 99% of my friends work for the federal government. So the question I get all the time is, why do I work with fungi? Luckily, the two fungal organisms that I work with are very interesting from a number of point of views. Some of them are called microsporidia, so these are important because they are obligate aggressive parasites of many animals, including human as well. So they're important from a veterinary perspective, medical perspective, and also from an ecological perspective, because virtually all animals are infected by microsporidia. And other ones are intriguing and important from an ecological perspective because they're involved in what we call the mycorrhizal symbiosis. So in these fungi associate with plants, and usually in their presence plants grow better, and therefore they're important from an ecological and agricultural perspective. So the talk today is divided into these two topics. I will first talk about Microsporidia and some of the past work we've done on these guys, and then we'll move on and discuss a bit more in detail the arbuscular mycorrhizal fungi, also called AMF in the field. So let's have a look first of all about the Microsporidia. So the first thing you should know about Microsporidia is that they are all obligate intracellular parasites of many animals, which means that in order to survive and propagate, they need to infect other plants or other animal cells. The animal cells that infect are of a wide variety and include also human cells. And in order to infect those cells, all AMF have evolved a very unique host invasion apparatus that we call the polar tube. In this picture here on the left, you can see there's two spores of a Microsporidia. I think the Microsporidium here is Nosema, but I'm not sure 100%, but it's a Microsporidium anyway. And you have two different spores here, one of which has the polar tube everted. So this is the polar tube. Imagine the polar tube as a harpoon, or actually a straw, a very long straw, that the Microsporidia will shoot at the host cells that they want to invade. That can be an animal cell, for example. And if the Microsporidia touches, if the tube touches the host cell, the entire content of these pores, so entire cytoplasm, will flow throughout the tube into the host cell and then start the parasitic life cycle. So it's a very mean kind of organism. It will shoot at you and then start dividing into your own cells. There's many genomes that are currently available. When I started my postdoc, there was one genome. Now there's about 30 or more different genomes. And there's one thing that we learn from these genomes is that it doesn't matter what species you look at, it doesn't matter what genera, they're always very reduced in content, which means that what I mean by this is that the genome have very few genes. And in some cases, the genomes can be extremely small. The smallest genome for a eukaryote is actually a Microsporidian genome, and it's about 2.3 megabases, so 2 million base pairs. It's very small, very compact. So why are they so small? Well, look at these guys in the cells. So this is a Microsporidian cell within the cytoplasm of a host. This is the Microsporidium, this is the nucleus of the Microsporidia, and all around it, this is the cytoplasm of the actual cells. It could be an animal cell, for example. Can someone here tell me what these different dark blobs could be? Exactly, these are the mitochondria. But these are not the mitochondria from the Microsporidia. Okay? It doesn't have a mitochondrion anymore. It lost the mitochondrial genome. This is mitochondria from the host. So imagine these guys, how mean they are. They will shoot a harpoon at you. They will transfer themselves into your cell, then surround themselves with mitochondria, and then start pump everything they can from the cells, including ATP, including amino acids, including nucleotides, folate, anything they can. So they're very lazy in some ways. And this kind of laziness have led their genome to reduce quite a lot. So this is just a diagram that shows an ancestral genomic structure. So this is your typical fungal genome when you have many genes that are separated by very large intergenic regions. And what happens in most Microsporidia, actually in all Microsporidia, sorry, is that most of these genes have been lost. Okay, so we have very few genes here. And in some cases, the loss of genes have been paralleled with also a loss of intergenic regions, which led in some cases to genomes that are not only gene poor, but also very small and compact, so very compressed genomes. So throughout my postdocs, which started in 2006 at the University of British Columbia, I tried to understand what are the consequences of this compression. Okay, when you have a very compressed genome, that will affect many processes, many cellular processes that are usually very important and very conserved among the eukaryotes, and these are the type of processes that are affected by this compression that include transcription of mRNA, the rates of gene rearrangements, and of course the host dependence. If you have very few genes, you cannot produce proteins anymore, so you rely a lot on the host for survival. Okay, so this is what I'm not going to talk about this in detail because this is very past work. This work that's been done almost 10 years ago. What I'm going to show you today is what happens when I started my tenure at the University of Ottawa. And when I started my tenure at the university, I started to sequence more genome. And what I found out is that actually it's not all about reductive processes in Microsporidia. Microsporidia are not only small organisms that are kind of boring to look at, very small genome, but when you have situations like this, when you have a cell within the cytoplasm of a host, the genetic content of this cell, in this case the nucleus, is also in close vicinity to the nucleus of the host. And that, in many cases, have led to a transfer of genetic material between the parasite and the actual host, okay? So in this case, we call this type of transfer horizontal gene transfers, and the definition of horizontal gene transfer is the non-sexual exchange of genetic material between two organisms that are present within the same environment. Those two organisms can be very different from one another. And in this case, I'm going to show you a transfer that goes from the microsporidia to the host, and in some cases to the host to the from the host, sorry, to the microsporidia. So we're going to show you now with some examples, which I think are striking examples of these type of transfers that can happen. The first transfer that we found has been found by my first graduate student, Mohammed Salman. And when Mohammed Salman started, I literally gave him two microsporidian genomes to annotate and to compare. These were the microsporidian genome of Encephalitoson helem and Encephalitoson Romali. Encephalitoson helem infects humans and vertebrates. E. helem infects insects. So I gave him these genomes. I asked him to annotate those genomes and to see whether he would find something interesting. And after about 2 months, he came back to me and he said, well, Professor, I found two interesting genes in there that are not supposed to be there. One of them is actually involved in the salvage of purines. And what he found out is that this particular gene is actually probably coming from an insect. So if you take those genes from the microsporidia and you do a phylogeny, those particular genes go and cluster themselves with very, very strong support in a clade that is only composed of insects. So when he first found that, I thought that this was probably a contamination, okay, because we obtained the DNA from the actual insect DNA, or from the actual insect and microsporidian DNA. But we found out that this is actually really within the genome. We confirmed that through PCR, so it's really genes that are very closely related to insects, but actually present in the microsporidian genome. What Mohammed found is that there are actually many other genes in these two species that are not present in any other microsporidian, and every single one of those genes is pretty much related to the metabolism of folate. So I would like you to focus on just this particular metabolism. Imagine that you work on microsporidia, and you're interested in knowing what they can do with the folate metabolism. You quickly realize that they cannot do very much. All they can do is steal folate from the host, thanks to a specific transporters that it's encoded within their genome, and then they use other genes here to feed this particular cycle and then use folate for a number of different things. What we found out is that the two species that I was telling you about a second ago actually can do much more. In this particular case, E. helem has many, many more genes here that are not present in any other microsporidia. So these are genes that are only present in E. helem and in the other closely related species. And every single one of these genes is actually related to this metabolism. So that tells you two things. First of all, that E. helem has I would say, a more sophisticated metabolism when it comes to that particular pathway. But it also tells us that these different genes must come from somewhere. Okay, they were either there ancestrally and then lost, or they've been acquired from other sources. And what we found out is that every single one of these genes are coming from different sources. These two genes are very, very similar in sequence and also in structure with genes that are actually from animals, in this particular case insects, and all the other genes up there are actually coming from bacteria. And we know this because these particular genes are only found in bacteria. They're not found in any other microsporidia and in any other animal. So this is the first case when you have a stepwise addition of different genes, piece by piece, in one particular species that ultimately leads to a better metabolism, in this case the metabolism of folate. So this is what happens in E. helem. I was telling you that there's another species that is closely related that we also analyzed that's called Iromalae. So these guys infect vertebrates, these guys infect insects. And what we found is that every single one of these genes that you see here are also present in both species, but all the genes that are involved in the folate metabolism in Iromalae are actually lost. They're there, but they're pseudogenized. So what that tells you that every single one of these genes have been acquired by the ancestor of these two different species, but they got lost in one and maintained in another. We don't know why, but this is what happens when you look at the actual genomes. A third case, I would say a third group of of genes or genomic regions that are often involved in this type of transfer between microsporidium and their host are the transposable elements. So the first thing that's interesting about the transposable elements, and that is almost counterintuitive, is that transposable elements can actually be very abundant within microsporidium genomes. In some cases, you find microsporidium genomes that have up to 270 families of transposable elements, and these 3 families are in 3 classes, which are your typical classes. You know, there's different kind of transposons, there's the retrotransposons and also DNA transposons. In the case of the DNA transposon, what happened usually, so these are genes which probably know that have a tendency to be present in a genome, then they excise themselves from one regions and they go in another portion of the genome. But what we found is that sometimes these different genomes don't jump between regions of the same genome, but they sometimes jump between the genome of a microsporidium into the genome of a host, and sometimes from the microsporidium from the host back to the microsporidium. Why do we know this? Well, we find many examples where you find transposable elements within the genome of a microsporidium that are almost completely identical to the same genes, but in this case, in the host. We found 5 cases like this, when you have actually an identity that is up to 99%. The cases that we found always involved microsporidia that infect honeybees. So this is the microsporidium. In this case, we found transposable elements in the microsporidium genome that are almost completely identical to the same transposable elements in the host. And the same case has been found also between the microsporidium that is infecting the pine beetle. And again, in this case, it's not a contamination. We went back to the genome. We confirmed everything through PCR. We confirmed everything manually by looking at the assemblies and so on. So this is really an event that is probably happened very, very recently, because the sequences are almost completely identical. Another interesting thing about this horizontal gene transfer is that every single horizontal gene transfer, not only microsporidia, but also in many other parasites that have been found to date, I always assume that the microsporidia or that the parasite is always the recipient of these genes. Okay, we never found a case where the parasite actually is giving genes to the host. And we found one example in the microsporidia where the host, in this case a planaria, has actually received genetic material from the parasite. We only found one case, and in this case we found that the genome of a planaria harbor many, many transposable elements that are only found in microsporidium genomes and nowhere else, which is an excellent case that proves that probably these genes are coming from a microsporidia. So if we conclude this first part of the talk, I hope you agree with me that microsporidia are not only these dull and reduced entities, but sometimes their genome are also quite malleable. They can move around, and through a horizontal gene transfer. These horizontal gene transfer are actually quite rare. We didn't find many cases for this type of events, but when they happen, they can also affect metabolic pathways, and we found in many ways, we found events that such event can actually help the metabolism of these parasites. As I said, these AGT events are generally very rare, but we found also evidence for some that are very recent. Okay, in these cases, when you find almost 100% identity between transposable elements among distantly related species, and in some cases bidirectional. So the microsporidia can also provide genetic material to the host. And this is quite intriguing because it suggests that perhaps these microsporidia, as they infect many different species within the environment, they can play a role as a vector of genetics, as a genetic vector for transposable elements to expand and move within particular environments. Do you have any question now for the first part of the talk? We can move on? Okay. Yeah? It's causing a disease that's called microsporidiosis. I think it's I don't know if you heard about toxoplasma, for example. Toxoplasma, it's a pathogen that's not related to microsporidia at all. It's an apicomplexan, and is very asymptomatic. Okay, and we found that the vast majority of the population has antibodies for toxoplasma. It's probably the similar case here. Okay, there are organisms that, unless you are severely immunocompromised,
Okay.
you have no risk. You're
Okay.
fine with it, and you can survive with no problem. So, I can tell you a funny fact is that I collaborate with a bunch of people in the Czech Republic, who actually the people provide me most of the time with spores, and it turns out that every single member of their lab has been infected at one point in time by Microsporia.
It's good to hear. It's good to hear that we are protected against.
Yeah, it's not a risk unless you are severely immunocompromised. Can I move on? Okay, good, fine. I just want to transition because I'm going to talk about something completely different now. So I've been talking about these nasty little parasites that, play around with the host in different ways. Now I'm going to talk about not parasite, but important symbionts, which are called arbuscular mycorrhizal fungi. So the 3 points I would like to make here are these ones. The first one is that arbuscular mycorrhizal fungi are not parasites. They're very important from an ecological perspective because they're involved in what we call the mycorrhizal symbiosis. In the symbiosis, the AMF will attach to the roots of most land plants and would help the plants gather more nutrients from the soil, such as phosphates, for example, or nitrates. They don't do that for free. They would expect the plant to provide back some sugars and lipids. Okay, so it's a symbiosis that goes both ways in this particular case. In the presence of AMF, generally plants grow better, but I would say also the ecosystem are generally more diverse, and therefore they're assumed to be very important from an ecological perspective. They're also very intriguing from a cellular and morphological perspective, or molecular perspective, if you like, because they are perpetually cenocytic. Perpetually. So what I mean by this is that if you look at if you open one of these spores, for example, or if you look at the hypha here, you'll see that there's absolutely no cell separation. So at all times, these organisms are multinucleated. We have from hundreds to thousands of nuclei that float within the same cytoplasm at all times. There's not a single stage where there's one nucleus or two nuclei. There's always thousands of nuclei within the same organism floating around in many different directions. The third thing that I would like to point out is that they're very difficult to work with, and because they're very difficult to work with, there's been a lot of controversies in the field. Okay, there's many results that I found not necessarily to be true or difficult to interpret and so on. My lab has been tackling two of these controversies. There's many, we tackled two of them. As you may have heard, AMF have been suggested by many to be ancient asexual organisms, and what I mean by this is that there are people who believe that AMF have evolved for almost 500 million years without any sexual mean to produce genetic variability. And what I mean by sexual here is no absence of mating and absence of meiosis. And for that particular reason, they're proposed to be evolutionary scandals. And other people have found some evidence that these different genetic nuclei that are present within a cytoplasm could be very divergent from each other, from one another, to the point where AMF may not have a genome, but might have actually a metagenome instead. What I'm going to show you today is that when you start gathering genome data from these guys and you start to analyze this genome data in proper ways, you quickly found out that AMF may be a little bit more conventional than previously assumed. And I say a little bit more because they're still kind of crazy, but not as crazy as people suggested earlier. And what I mean by this is that they probably undergo sexual reproduction on a regular basis. You just don't see it because it happens on the ground. It's cryptic, so you don't see it. And these different nuclei, I'm going to show you that these different nuclei, I'm going to show you some evidence that these different nuclei that are present within the same cytoplasm are either completely identical to one another, and in some cases, there's two kind of nuclei, a population of two diverse nuclei that are present within the same cytoplasm. So all the data I'm going to show you today is based on 5 isolates, so 5 single spore cultures that have been harvested about 10 years ago in a field in Switzerland by my former colleague Alexander Koch. So what Alexander Koch did, he went to Switzerland in a field, collected single spore of the same species. The species here is called Rhizophagus irregularis, brought the single spores back to the lab, produced cultures from them. Okay, I got the cultures, we propagated the cultures, we sequenced the DNA from those cultures using Illumina paired end and med-pair, and then we assembled everything together into genome assemblies. Now I'm going to show you some boring genome assembly data. Here is exactly what you would expect from a genome assembly. So don't look at all these numbers. I would like you to pay attention to what I think the most important column is, which is the last one. So this last column here show you the number of SMPs per kb. So the number of SNPs per kb is a measure that gives you an overall overview of the diversity, genetic diversity that is present among these different nuclei. The genetic diversity here that you see is actually not very high. What I can tell you is that if you can do exactly the same analysis on our genome, you can do the same analysis of any conventional fungus, you will find values that are very, very similar. So that tells you already that the genetic diversity is not unusual when you look at the high level like this. What we found as well is that there's actually divergence and difference in the genetic diversity between these different isolates. So in this case, for example, the isolate A4 and the isolate A5, these numbers are isolates, okay? So they're single spore cultures. So it's one individual. Imagine these are different individuals of this particular fungus. So you can see that these are actually, they have a genetic diversity that is double or more than double of the other isolates. So the first thing that we're trying to understand is where this increase was coming from. And what we found out is that isolates that have a higher genetic diversity tend to be heterozygous. So this is just to show you one specific region of the genome of some of these heterozygous, of some of these isolates with higher genetic diversity. You can do the same analysis on any other genomic region, you will find exactly the same thing. So in this region, you have about 5 SNPs here. This is 1, 2, 3, 4, 5 SNPs. And you can see that the SNPs are always biallelic, first of all, and they're always at a 50-50 ratio. And what I mean by this is that half of the reads that are plotted, that are mapped here are a T, and the other one are a C. Here, the half of them are A, half of them are G. This suggests that there's some heterozygosity going on at that particular region. Okay? And if you do the same analysis across the entire genome, as opposed to just one particular region, you find exactly the same thing. The vast majority of the allelic variation that you see, it's at a 50-50 ratio here. And if you do the same analysis on your genome, you will find the same thing, pretty much. So what it tells you at first, that perhaps those two isolates that have higher diversity are actually heterozygous and possibly diploid, while the other ones are homozygous and haploid. Now, the good thing about ploidy is you can actually test it. You can try to do flow cytometry to determine the difference in ploidy between the different individuals, which is exactly what Kinga has done. Now, what Kinga has found is that it doesn't matter whether you look like a diploid or you look at like an haploid, you're always an haploid. I know it's kind of complicated, but it's a bit of a conundrum. But imagine that you have an heterozygous peak like this, you have actually an haploid genome. You have an homozygous peak like this one, you also have the same type of haploid genome. So it's a bit of a conundrum here. How can you be heterozygous and at the same time being haploid? The only thing that came to my mind is that perhaps these two guys here that are heterozygous are actually dikaryotic or dikaryotic. And what I mean by this is that within the population, within the mycelium of this guy and these guys, you have a population of two distinct nuclei pretty much at the same ratio that are wandering within the same cytoplasm. It's the only explanation that I had at the time. So if this crazy hypothesis is true, that would be something completely new, by the way, that has never been found anywhere else. So if this crazy hypothesis is true, we should be able to get some single nuclei from these guys here and these guys here and sequence them and find two different kind of nuclei. And you should do the same thing for this guy and find just one single type of nucleus within the actual isolate, which is exactly what Kinga has found. So if you look at these two isolates, A1 and C2, who don't have that heterozygous peak, you find that each one of the nucleus here, so we took nuclei and we sequenced the nuclei, we genotyped them at different regions, okay? And we find there's actually one genotype. Each one of the nucleus here, it's a different nucleus, has the same genotype. Same thing for A1 and same thing for C2. Of course, they have different genotypes because they're different isolates. If you do the same analysis on A5, which is a heterozygous looking isolate, you find actually two kinds of genotypes. Some nuclei have the genotype here that is the dark green genotype, and other ones have the light green genotype. So this seems to prove the fact that there are two kinds of isolates. Some that are, I would call them homokaryotic, but all the different nuclei that are present within the same cytoplasm have the same genotype or very similar genotype, and other that are dikaryote or heterokaryote, if you like, depending on their definition, where you have actually two kind of nuclei present within the same cytoplasm. So the good thing about this homokaryotic, dikaryotic genome organization is that Dikaryosis, something that is similar to this, not exactly like this, but less complicated, actually exists in other fungi. Dikaryotic fungi exist out there, and it's always a result of sexual reproduction. In dikaryotic fungi, you have also two homokaryotic fungi that get together and then exchange themselves nuclei to form a dikaryon, a cell that have two compatible nuclei at the same time within a cell. Okay, so if these guys are really dikaryotic or behave like dikaryotic fungi, you should find within this genome, you should find a region that we call the mating type locus. Mating type locus is a region that control sexual identity in fungi, and two organisms that have compatible mating type loci get together and undergo sexual reproduction. And if it's true, the region not only should be there, but it should be there in two copies, and these two copies should be very divergent from one another. And this is exactly what Jeanne has found. And she found that in this particular region for the isolate A5. So this is just a figure that shows you the coverage along one of the many contexts that we have. So here, the black line here is actually the coverage along this region. It's about 80x, pretty good coverage. And you will see that the coverage here drops exactly at 50%. I'm not making the number up. It could be 49, but it's very similar to 50%. Okay? So, and if you look at this region, it turns out that that region harbors two genes that are found in another scaffold of exactly the same size. Well, there's another scaffold that is divergent, that has exactly this size, and both of these harbor two homeodomains which show a lot of similarity both in structure and in sequence with the mating type locus of basidiomycetes and some ascomycetes. So what does this locus look like? It looks like something like this. Okay. Along this drop in coverage, you will see there's actually two different kinds of genes. Okay, these are the genes that we think are part of the mating type locus of the arbuscular mycorrhizal fungi. I would like to point out that this region shows a lot of strong similarities with the mating type locus of the basidiomycetes. It's composed of two genes which are bidirectionally transcribed. Both genes have a homeodomains. They also have a coil-coil motif, which is essential for the proteins once they're transcribed to get together and form a heterodimer, which is essential for sexual reproduction to occur. And it also has nuclear localization signals, which are also essential for sex to occur as well in this fungi. Most importantly, these are present in two copies in the dikaryon A5 and A4, one copy per nucleus. Okay, you never have both copies in the same nucleus, and in homokaryon only have one copy. Okay, and it's interesting to see that some of the copies here present in some of these homokaryon, here the different colors actually show sequence identity. Okay, so if you have the same color, it means that your mating type locus is almost identical. And if you look at this, A5, the copies that are present here are actually identical of the copies that are present here and here, which suggests that perhaps A5 has been formed through the fusion or genetic exchange or nuclear exchange between something that resembles A1 and something that resembles C2. So if you add A1 to C2, you get A5, basically. That's what I'm trying to say. One last thing that I would like to point out is we will try to also understand the evolution of this particular locus. And to understand the evolution of this locus, we decide to amplify and isolate this locus from about 30 plus isolates or cultures that we have in the lab. And the results of this isolation is something like this. So this is the phylogeny of those particular two genes obtained from many different isolates that we're growing in the lab. This phylogeny is interesting from three particular reasons. First of all, it gives you an overview of the diversity of the mating type locus. And you can see that this locus actually segregates in six very strongly supported clades, which could or could not be different mating type, which are involved in compatibility in AMF. The second interesting thing is that when you have something like this now, you can go back to the phylogeny and see how the dikaryon have originated. All you have to do is to go and seek for the mating type loci that were present in the dikaryon. So let's do that together. Let's see, for example, let's try to see let's try to find the two mating type loci that are present in the dikaryon A4. You find out that actually here, there's one right here, and there's another one right here. So these are the two genes that are present within the dikaryon itself. And what tells you possibly is that the AMF A4 is actually originated from two homokaryons that are closely related from an evolutionary perspective. If you do the same exercise from other dikaryon, for example A5, in this case you find that in some cases some dikaryons have originated from quite distant isolate. In the case A5, and you also find a third dikaryon. Actually, we find more thanks to some collaboration with Frank. But for now, I'm just doing with these three ones, which we call SL1, which appear to be intermediate between these three. So what we're trying to do now is to use these different clays to possibly reconstruct dikaryon in the lab. For the first time, we could theoretically be able to produce new genetic strains of this AMF, which is something that was not feasible before this data. So if we take all the data that I show you until now, if we put that all together, our lab was able to build the first model for the biology of the Bruschka mycorrhizal fungus. And the model suggests that AMF undergo two life stages. A life stage that is kind of boring and clonal, which is the stage that everybody knows about, which is this one where every single mycelium of an AMF have many nuclei that are almost identical or completely identical to one another. And in some cases, our model suggests that this, at one point in time, for a reason that we still don't know, two AMF can get together and through compatibility can exchange nuclei to form what we call a dikaryon-like or heterokaryon stage when you have at this point two different nuclei within the cytoplasm. If this hypothesis and model is correct, you would expect the dikaryon to undergo karyogamy. So these different nuclei should fuse with one another, become diploid from haploid, and then undergo meiosis or parasexuality to create new divergence in this case. Okay. What we know so far is that, so what I've been showing you and what's been published so far is this, that this stage exists and this stage exists. We still don't know how to get from this stage to this stage, although we're making progress from that point of view. And we recently found out how this stage works. We found out there's actually a lot of recombination happening between these different nuclei. This is unpublished, so we'll ask you not to talk too much about it or tweet about it, because this research field is crazy. So keep it up for yourself. I'm going to show you some of the data. So remember when I was showing you these different genotypes of the single nuclei that you got? So we did exactly the same exercise, but instead of genotyping these nuclei, we sequenced a whole bunch of their genomes, and we compare these different genomes together. So this is unpublished, so it's not super high quality figure, but so these are the two dikaryons I was mentioning for a while, okay? And these are all the nuclei that we actually sequenced. So we sequenced a whole bunch of the genome of each one of these nuclei, and then we compare their genotypes. So if nuclei have the same genotype, they have the same color. So what that tells you is that in A4 and A5, you have a lot of structure. And usually, when you have a specific mating type, here for these three nuclei, we know the mating type of this nuclei. For this nuclei as well, we know the mating type of this nuclei. We don't know the mating type of this nuclei because we don't have enough DNA to go back and check for the mating type. But there's a lot of structure going on. And what that tells you is that there's always a strong correlation between the mating type and the genotype. Okay? This is what happens in A4 and A5. Remember, I tell you we found another dikaryon, which is called SL1, and in SL1 we don't find this at all. It's a mess. Okay? So we have about 16 different nuclei here that we sequenced, and there's absolutely no correlation whatsoever. It's a genomic recombining mess. Okay? There's no correlation between the mating type and the genotype whatsoever. And when you look at the actual sequence, you will see that those nuclei recombine like mad. We don't know whether meiosis or parasexuality is involved in this, but what I'm telling you is that basically what this shows you is that AMF have found ways to create new genetic diversity without observable sexual reproduction. So this idea that they're ancient asexuals, forget about it. Another interesting thing is this. When I show you a picture like this, it gives the impression that there's actually a lot of diversity going on. But the point I'm trying to make now is that when you actually look at the entire sequence of these different nuclei and you compare them, the amount of variation is extremely, extremely small. So all these nuclei that are present, they're always very, very similar to one another. But when there is variation, it's either structured or is highly recombining. But the variation overall is very, 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 very low. So to summarize this, our data suggests that AMF undergo two life stages, homokaryotic or dikaryotic. We also found what I think is the best candidate by far for a mating type locus in the AMF. There's one thing I would like to point out is that the locus that we found shows striking similarities with the mating type loci of sexual fungi. But we did not prove that this is actually a mating type locus functionally, okay? But it quacks like a duck, walks like a duck, is really, really strongly looking like a duck. But we did not test it functionally. We found evidence of a lot of internuclear recombination in some dikaryotic isolates, and I think that we definitely found the source of genetic variation. So if people are interested in finding new genetic diversity in AMF, they have to look at dikaryotic isolates. If you look at homokaryotic isolates, you find no diversity whatsoever. How much time do I have left? Can go ahead? Yeah? Okay. It's almost over, don't worry. So a third thing, actually, something else that's quite interesting from a biological perspective with AMF is that they also harbor quite a lot of intraspecific phenotypic variation and genotypic variation. And what I mean by this is that imagine that you go in the field and you start harvesting single spores of the same species from the same population. You bring back these spores in the lab and you start cultures from these spores. You will see that these cultures are actually quite divergent between one another. So you have some cultures that produce a lot of hyphae and very few spores, other cultures that produce a lot of spores, very dense, but very few hyphae and so on. And this intraspecific diversity is quite important because it has been shown to be correlated with variation in plant growth. So what I'm telling you is that if you take 3 different spores of the same species from the same population and you put them into symbiosis with the same plant, the plant will grow in completely different ways. So this variation is important from a phenotypic and also from a agriculture perspective. So what we tried to understand recently is what's creating this phenotypic variation at the genome level. And in order to understand and address these questions, we went back to NCBI. We gathered all the genome data that was available at the time. So these are the 6 assemblies that I was telling you about a second ago. So we took these different assemblies, we annotate every single one of the genes in there, and we compare the gene repertoire between each one of these isolates. And what we found is a huge amount of diversity, completely unexpected. So in this particular figure here, I show you, so this is the phylogeny of the 6 isolates, so the 6 single spore culture of the same species for which a genome is available. We annotated each one of these genomes, and this is the abundance of genomes that are shared between these isolates and non-share, that are unique to each one of these isolates. So the point I try to make here is this, is that the amount of genes that are shared between each one of these 6 isolates are actually present in dark blue and in light blue. Everything that's beyond that is variation. Okay? So these are all what we call accessory genes. So what I'm showing you here is basically that half of the gene repertoire of these 6 isolates are variable in presence and absence and in copy number. So there's a huge amount of functional diversity in each one of these different isolates. And just to give a bit more of an idea of the variance, we have almost a fifth of the gene repertoire that is isolate specific. So here I'm talking about genes that are present in only one isolate of a species and absent from all of the isolates of the species. And you also have a substantial variation in FAM distribution. So these are motifs that give function to a protein. Okay? So we have some isolates that have, let's say, that are highly enriched for certain motifs, and those same motifs are completely depleted in other isolates of the same species, which suggests that there's a lot of variation in the molecular function of each one of these isolates. And so the reason why, we don't know, but it's probably because this is helping them adapt to different environments. And maybe the reason why they're so ubiquitous, maybe the reason why they can associate with any kind of plant. So it gives them more opportunities for adaptations, if you like. So this is what happened in the case of genes. But the same pattern happens also at other levels, in this case in transposable elements. So the 3 points here that are important is the first one is that transposable elements are extremely abundant in AMF genomes. They make up almost half of the genome is transposable elements. So there's a lot of them in the genome itself. The second point is that they're also very different between the different isolates, where you have some families or subfamilies of transposable elements that are very abundant in some isolates and actually completely absent, in other isolates. So there's a lot of variation in the number and structure of these transposable elements between the isolates of the same species. And most of them are expressed. We found that almost 90% of these transposable elements are expressed within the genome, which means that they're active. And that means that they're moving around quite frequently. And as they move around, they probably create a lot of new genetic diversity. They break things, they change structure, and so on. They produce rearrangement and recombination. One very last thing I want to talk about is this, is that when you look at this genome, that are supposedly of the same species, you find a lot of what I would call phylogenetic incongruence. And phylogenetic incongruence is some conflict between phylogeny and what you see at the level of the genome. So imagine that you take the mitochondrial genes, or you take, for example, the ribosomal genes, or in fact, any kind of genes from these different isolates, and you use that to produce a phylogeny of this isolate, you always see this type of phylogeny, always. You always see A1 that is closely related to the isolate B3 and A5, and then you have the other isolates that are kind of more divergent. But when you look at the genome of these guys, you often find this, but many times you don't find this type of structure at all. In this particular case, this is a region that is about 60,000 base pair in length, that is actually syntenic between each one of these isolates. Every single gene that is in red here is a gene that is syntenic. It means it has the same order and same direction in every single, and it's present at the same location in every single one of these isolates. And you can see here that the isolate A1, which is supposed to be closely related to B3 and A5, has actually the same exact structure as C2, which is an isolate that is much more divergent. And you find the same kind of phylogenetic inconsistency across the entire genome, and it doesn't only affect one isolate, it affects many different kinds of isolates. And that suggests that it's probably the result of past gene exchange or genome exchange or rearrangement between the nuclei of different isolates in the field. So to summarize and conclude here, the last part of the talk, I'll show you that R. irregularis, which is the model AMF species, so these are the isolates I was telling you about pretty much the entire time, harbor a ton of accessory genes, and this high genome diversity is probably the result of high paralogy and transposition in these organisms. We don't know why there's so much diversity at the genome level, but it's probably helping them from an adaptive perspective. And we also find a lot of evidence for past inter-isolate genetic exchange. We don't know whether these exchanges between isolates are driven by sex in this case, or by horizontal gene transfer. But hopefully we'll find out one day. So with this, I would like to, I hope I didn't bore you too much, and I would like to thank you for your attention. This is some of the institutions that have given me money for this type of analysis. This is my lab. I'll show you a bit of a past lab, but I like this picture. And these are some of the collaborators that have helped me produce and analyze some of the data I showed you today. So with this, I thank you, and I'll take any questions.
Bien, merci Nicolas pour cette brillante présentation. Et en passant, ces travaux, Nicolas, il les a publiés il a été invité à faire une revue de littérature sur la génétique des champignons mycorhiziens arbiculaires qu'il a publié dans Trends in Plant Science, je crois. Donc, vous pouvez toujours y aller pour trouver l'information nécessaire. Donc, c'est la période des questions. Je vais passer avec le micro. Si vous me permettez, Christian, oui.
Merci pour ta présentation. J'ai une question sur les cas où tu vois des dicaryons, deux noyaux différents, puis tu montrais au départ que la fréquence des allèles était toujours autour de 50%. Il me semble qu'un cytoplasme avec deux noyaux, éventuellement, il va y avoir de la variation dans la fréquence, et de la dérive, de la perte. Ça me semble surprenant que ça reste autour de 50%. Est-ce qu'il y aurait une genre de sélection, une complémentation entre les deux, qu'il y aurait des allèles délétères qui les maintiendraient à une fréquence comme celle-là? Ou...
Donc, je ne sais pas vraiment. Okay? Ce que je peux dire, c'est ça. C'est que le 50-50, ce qu'on a trouvé récemment, ça n'a pas été publié, c'est probablement le résultat du fait que toutes les analyses génomiques sont faites sur de pardon, je vais recommencer depuis le début. Il est impossible d'obtenir de l'ADN, ou il est probablement possible là, mais il est très difficile d'obtenir de l'ADN d'une spore et d'analyser tout le génome. Donc, toutes les analyses génomiques qu'on a faites là, c'est à partir de plusieurs microgrammes d'ADN qui ont été obtenus à partir de millions et des millions et des millions de spores et des millions et des millions et des millions de différents noyaux. Donc, si ce n'est pas des milliards de noyaux. Donc, toutes les données ici, là, c'est une moyenne de plein de noyaux différents. Ce qu'on a remarqué, c'est que cette moyenne, c'est dû au fait qu'on a probablement obtenu les données à partir de plein de noyaux. Si maintenant tu fais le même exercice, mais à partir de single spores, on a remarqué qu'il y a une certaine diversité de spatiale. Où si tu prends des spores dans une certaine région du mycélium et tu fais cette analyse, tu n'as pas ce 50-50. Et dans une autre région, tu n'as pas ce 50-50. Donc tu as des régions qui ont tendance à avoir plus de mating type 1, d'autres régions qui ont tendance à avoir plus de mating type 2 et d'autres régions où tu as les deux. Donc je ne sais pas si ça répond à ta question, mais c'est du 50-50, c'est pas un artefact si tu veux, mais ça t'indique qu'en général c'est 50-50 si tu prends vraiment énormément de mycélium. Mais si tu regardes spatialement avec plus de précision, tu as plus de variations.
Une autre question? Oui.
Oui. Est-ce que vous trouvez des gènes de recombinaison méiotique dans vos génomes d'AMF?
Oui, tout à fait. Donc ils sont tous là. Ça, c'est le premier papier que j'ai publié sur les AMF en tant que, une fois que j'ai eu mon poste de prof là, où effectivement tous les gènes qu'on appelle qui sont essentiels pour avoir une méiose conventionnelle, ou théoriquement pour que les organismes puissent avoir une méiose complète, sont là. Y compris les plus importants, SPO11 par exemple, ou REC8, ça c'est des gènes qui sont généralement les plus importants, ou qui doivent être absolument présents, ils sont présents. Ce qui est super intéressant par rapport aux gènes de la méiose, c'est que certaines des critiques qu'on m'a indiquées à l'époque, c'est plus des critiques qui existent maintenant, mais c'est des critiques que j'avais à l'époque, c'est que c'est pas parce qu'ils sont présents qu'ils sont nécessairement impliqués dans la méiose. Ce qui est intéressant, c'est que quand on regarde la transcription de ces gènes, s'ils n'étaient pas impliqués dans la méiose, et s'ils étaient impliqués par exemple dans la recombinaison mitotique, tu t'attendrais à ce qu'ils soient exprimés tout le temps. Ce qui n'est pas le cas. Par exemple, si tu regardes le gène SPO11, par exemple, il est pratiquement, je crois qu'il n'est pas du tout exprimé. Ce qui indique qu'il est exprimé à certains moments critiques. Et ce moment critique, c'est probablement la méiose.
Bon, merci Claude. Et puis une autre question. Ah, Yves. Oui.
Bonjour. Je suis tellement content d'être venu ici parce que ça va me réconcilier avec les champignons domycorhiziens arbusculaires, notamment que les noyaux sont plutôt semblables que différents entre eux dans la même spore. Ma question s'adresse à ce que les biologistes connaissent depuis longtemps, la méiose. Comment on fait pour suivre la méiose chez un organisme et chez les organismes AMF, où pourra avoir lieu la méiose? Durant la formation de la spore, dans la spore ou lors de la germination de la spore?
Donc, je ne sais pas. Okay? Ça, c'est quelque chose qu'il va falloir trouver. Ce que je peux t'indiquer, c'est que, si je reviens sur les différents... une situation comme ça, là, où on a énormément de recombinaisons entre les noyaux, le montant de recombinaison t'indique une chose, c'est que ce n'est pas juste un événement spécifique. Quand tu regardes les noyaux, c'est pas... les événements de recombinaison ne peuvent pas être corrélés avec un événement méiotique. Donc moi, ce que j'essaie de dire là, c'est que s'il y a méiose, ça arrive à différents endroits dans le mycélium et c'est pas quelque chose qui est synchronisé. Et c'est pas quelque chose qui arrive nécessairement entre un noyau originel et puis ensuite ça continue.
Donc la mitose ne serait pas synchronisée non plus comme ça.
Peut-être pas, oui. Si la méiose n'est pas synchronisée chez d'autres champignons filamenteux, c'est très probablement pas synchronisé ici.
Ouais.
André, non? Christine, et puis je viens d'apprendre.
Oui, bonjour. Moi, ma question est un peu sur ton feeling, parce que tu nous montres qu'il y a beaucoup d'adaptabilité potentiellement de ce champignon-là, ce qui n'est pas étonnant vu sa biologie, qui est quand même unique au monde, là, et symbiote obligatoire. Fait que, est-ce que vous avez une idée, par exemple, il pourrait y avoir de la génétique qui s'exprime juste à la germination de la spore, avant le stade symbiotique, après le stade symbiotique, puis comment, au point de vue, est-ce qu'on a des outils pour analyser ça, ou est-ce que c'est bien trop compliqué, parce que tu dis qu'il faut des millions de noyaux, puis dans la technologie, est-ce qu'on va pouvoir un jour s'en sortir pour répondre à des questions de manière fiable?
Je pense que le seul moyen de répondre à ces questions, c'est de faire de l'expérimental, dans le sens où tu devrais, par exemple, idéalement... Ce qu'on essaie de faire maintenant, c'est de produire des nouvelles souches à partir de simples noyaux de ces dicarions. Et on sait que ces simples noyaux de dicarions, ils sont recombinants, donc ils n'ont pas exactement la même organisation nucléaire, ils sont recombinants. Donc maintenant, on essaie de produire ces souches, c'est pas facile, on essaie de les produire, et ensuite on va les analyser génotypiquement. Et ça va nous donner un peu plus d'informations pour savoir quand est-ce que la recombinaison arrive, à quel moment, quels sont les endroits où tu as plus de recombinaison par rapport à d'autres, etc. Le travail qu'on a fait ici, là, la chose la plus importante, je pense, de ce travail, c'est que la majeure partie, 90% si ce n'est pas plus, de toutes les données physiologiques, de toutes les données moléculaires, de toutes les données de génomique, etc., sont basées sur cette souche-là. Il y a une souche, en agronomie, c'est la même chose. La majeure partie des inoculums, c'est cette souche. Donc ça t'indique un petit peu le type de variation qu'on peut avoir à l'intérieur d'une espèce, quoi. Donc l'idée, c'est ça, c'est que tout ce qu'on sait sur une souche, ça nous indique, c'est une fraction de ce qui se passe à l'intérieur d'une espèce. Et ça, c'est le point important de ce travail.
Oui, Franck.
Tu sais, on a fait un criblage récemment de toutes les Rhizophagus irregularis qu'on a dans la collection. On a fait à peu près une centaine de souches, incluant celles que tu as présentées. Et on a trouvé des dicarions potentiels, peut-être pour 5 à 10 souches. C'est peut-être 5 à 10% des dicarions qui sont présents sur les souches qu'on a analysées. Donc je m'interroge sur le côté... est-ce que c'est quelque chose de vraiment prépondérant en nature? Est-ce que tu as des réflexions à ce sujet-là?
Bah, tu l'as dit toi-même, là, c'est 10% de souches, c'est probablement pas prépondérant. Et le problème, c'est que toi, les souches qu'on a, c'est des événements qui sont apparus dans le passé. Okay, on a isolé des simples souches, on a formé des cultures à partir de ça, donc c'est des événements qui... Mais pour répondre à cette question de manière définitive, c'est ce que j'aimerais faire maintenant, c'est aller dans les champs et investiguer la présence ou pas de dicariose selon différents types d'environnements, selon différents types de plantes, par exemple, etc., etc. Mais la fréquence, là, je ne sais pas. Mais clairement, il semble que ça soit rare.
André?
Si on effectue une culture monosporale, est-ce qu'on peut s'attendre, après repiquage et repiquage et repiquage, d'avoir une variation génétique?
C'est quelque chose que, probablement, au bout d'un moment, non. Justement, tu t'attends à ce que ça soit plutôt uniforme.
Mais ça n'a pas été fait encore, ça.
Ça a été fait, mais le problème, c'est que c'est des générations qui ne sont pas suffisantes pour voir une différence. Mais tu sais ce qui passe, hein, tu connais. Rien que pour commencer une souche, il faut 3 mois. Donc pour faire 1000 générations, bah, il faut 2 vies, quoi.
Donc de ce côté, il n'y a pas de questions. Je repars alors avec...
Il y a une question, Franck.
Oui.
Pour compléter sur ce qui vient d'être dit sur la question de Monsieur Fortin, il y a une publication qui est sortie dans Mycorrhiza cette année sur Funiformis mossé, qui a été cultivé pendant 20 ans en laboratoire, dans 4 ou 5 laboratoires différents en Europe. Donc c'est parti d'une spore. Puis ils ont regardé quelle a été la dérive génétique sur finalement les 5 souches qui étaient ensuite maintenues pendant ces années-là dans les différents laboratoires, et une dérive génétique qui a été observée. Elle n'est pas forte, mais elle est là, elle est présente. Et il y a aussi les données d'anastomose qui montrent que, évidemment, lorsqu'on essaye de recroiser ces souches-là entre elles, les taux d'anastomose chutent énormément, de l'ordre de 5 à 15-20%, alors que lorsqu'on recroise, on fait les croisements des différents réplicats de la même souche, ben là on a des taux d'anastomose de 40 à 60-80%.
Oui.
Merci. Est-ce que vous avez fait ou observé une différence de, je dirais, de comportement entre les dicarions et les homocaryons?
Non.
Non.
Non. Justement, ça, là, ce qu'on aimerait faire maintenant, c'est voir un petit peu par rapport à la plante, là, s'il y a des variations au niveau de la plante. Mais quand tu les regardes, même d'un point de vue du noyau, etc., tu vois la même structure de noyau, la même différenciation spatiale des différents noyaux. On n'a pas une structure, par exemple, pour répondre à la question que Christian avait auparavant, où est-ce qu'il y a, par exemple, 2 noyaux qui s'attachent chez les dicarions, puis pas chez les monocarions, on n'a pas du tout ça. Donc on n'a pas vraiment c'est pas facile à les distinguer. En fait, c'est pas du tout faisable.
Ah, Franck encore, oui.
J'avais une petite question, j'avais une petite commentaire sur sur le papier de mycorrhiza là. C'est que y a un petit truc qui me tracasse un petit peu sur le papier, c'est comment est-ce qu'ils peuvent s'assurer que c'était vraiment les mêmes souches. Ça fait 20 ans qu'ils les ont pas vues.
C'est des travaux publiés par Manuela Giovannetti, puis je pense que c'est elle qui a parti ça comme il y a 20 ans en arrière, donc j'imagine que le suivi était rigoureux là-dessus.
Non, je dis ça parce que c'est facile de changer les cultures.
C'est sûr. On l'a déjà vu dans le passé. J'avais une autre question. Oui, tu l'as évoqué au début de ta présentation, qu'il y avait pas mal de divergences, d'opinions divergentes par rapport si les noyaux sont génétiquement pareils ou différents. Et il y a quand même eu beaucoup de publications dans le passé qui ont montré que ces noyaux-là étaient différents. Pourquoi est-ce que maintenant, ces évidences-là semblent tomber, mais est-ce que c'est un effet juste de l'apport technologique qui fait qu'on a des meilleures données et à ce moment-là, on a une vision qui est vraiment plus précise de la diversité génétique des noyaux? Est-ce que c'est vraiment ça qui fait qu'on tend maintenant vers ce modèle-là?
Donc, j'ai pas envie trop de parler de ça là parce que j'ai fait mon doctorat dans un laboratoire qui était très fortement, ou qui s'est bâti sur cette histoire d'hétérokaryose là. C'est bâti sur cette histoire que ces noyaux étaient très différents les uns des autres. Je pense que le problème c'est ça, c'est que pendant très longtemps, des personnes dans certains laboratoires ont utilisé des données et des outils qui peuvent être interprétés de 5 manières différentes. Mais leur interprétation était basée juste sur l'hétérokaryosie. Donc des résultats, par exemple, qui démontraient la présence de variations entre noyaux, auraient pu être interprétés par la présence d'artefacts, ou la présence, par exemple, de variations à l'intérieur du génome, etc. Mais la seule interprétation que ces personnes ont donnée, c'est l'hétérokaryosie. Donc c'était un petit peu une idée au lieu de parler d'autres interprétations, ils ont donné seulement leur interprétation qui, à mon avis, était fausse, qu'on a démontré était fausse.
Ok, Agathe, peut-être on va aller vers la dernière. Un instant.
Est-ce que justement, toi, tu n'interprètes pas plutôt c'est volontairement identique? Est-ce que maintenant, justement, il y a une progression au niveau technologie, puis les outils qui permettent de dire ben moi, je me suis enlevé tous les biais, donc.
Oui, tout à fait.
C'est
vraiment dans ce sens-là. Mais justement, c'est pour ça que j'ai pas envie d'en parler, parce que il est vrai que les outils dont moi j'ai accès de nos jours, c'est aussi des outils qui sont accédés par tout le monde. Hein, je les ai pas inventés ces outils. Mais pour moi, il est important, et je pense qu'il y a quelque chose que quand je donne différentes présentations, là, j'essaye d'expliquer le plus possible. Par exemple, quand on a trouvé le Metintaplocus. Ce Metintaplocus, je ne sais pas, mais c'est ça Metintaplocus. Mais je suis honnête en disant que il ressemble à 100% à Metintaplocus, mais on n'a toujours pas fonctionnellement prouvé que c'en était un. Donc c'est comme ça que la science doit marcher. Et malheureusement, dans le domaine de l'AMF, ça n'a pas été le cas pendant très longtemps. Il y a eu cette idée, ces limites idéologiques. Et ce que j'essaie de faire dans mes revues, par exemple, c'est d'expliquer ça de manière polie et d'expliquer qu'effectivement, c'est avec l'avancée de... Ouais. Tout à fait, ouais.
Bien, alors je crois qu'on a épuisé toutes les questions, alors j'aimerais que vous puissiez me joindre à moi pour... Bon, alors, quelques annonces. Donc, nous aurons encore une conférence CEF-IBIS ici, le 16. Que la conférence sera prononcée par le professeur Yousri El-Kassabi, qui vient de UBC. Il va nous parler de la génomique, de la sélection génomique des arbres forestiers. C'est un forestier, un généticien forestier que Armand doit connaître, je crois. Donc, c'est le 16 à 15h dans cette salle. Et puis, l'autre annonce importante, c'est évidemment le colloque CEF. Du 30 mars au 1er avril, on a la conférence du CEF, donc, qui est un regroupement stratégique financé par le FQRNT. Alors, c'est Laval qui va accueillir le colloque CEF cette année. Donc, je vous invite à vous joindre à nous pour ces colloques-là. Encore une fois, merci d'être venu en si grand nombre et je vous souhaite un bon lunch.